I think there's an increasing recognition that biofilms play a role in chronic diseases. And the reason for that is that biofilms are very difficult to eradicate. They live on surfaces. They produce extracellular materials that can defend themselves against human immune cells or antibiotics. And therefore, they're very difficult to remove. And once they form on a surface, they're very difficult for your body or for drugs to kill them.
So it's only been within the past really 15 years that the medical community has recognized the existence of biofilm bacteria as fundamentally separate and distinct from planktonic bacteria.
Bacteria come together when they're threatened. When you have a situation where bacteria are treated with antibiotics that can kill them, or when a strong immune system responds to bacteria by producing cytokines or chemokines or other agents that are able to destroy the bacteria and to eliminate them from the body, these bacteria have a choice. They can either stay as an individual bacteria and die, or they can change their form, and instead of being a planktonic bacteria, form biofilms.
Acute bacterial infections and chronic bacterial infections are very different. Acute bacterial infections are caused by planktonic bacteria, that is, bacteria that are swimming freely. Each bacteria is essentially a single-celled organism. Chronic infections, for the most part, are caused by bacterial biofilms, and the bacterial biofilm is, in essence, a multicellular organism, so that each individual bacteria within the biofilm is analogous to each individual cell in your body, and so that they are actually functioning as a single organism. And it's the adoption of the biofilm mode of growth that provides for what we say chronicity or persistence.
We have done studies in which we have grown biofilms and then tried to add, penicillin, gentamicin, some standard type antibacterial agent, and we're not seeing it do anything.
They are very difficult to eradicate because they don't grow rapidly, and most antibiotics kill organisms that are growing, and they will not kill organisms that are not growing. So these organisms can fester in a particular area. For example, a heart valve endocarditis is a biofilm of organisms on that heart valve.
Examples of this can include infections of bone, infections of your heart valves, infection of the urinary tract, in which bacteria can colonize these surfaces in the human body and live there for long periods of time.
And what we know about these communities is that it takes such a greater concentration of antibiotic to get rid of them that it is not indicated. You're talking about anywhere from 500 to 5,000 times the concentration that normally is the minimum inhibitory concentration of antibiotic that it takes to kill bacteria in a biofilm.
The link between biofilms and antibiotic-resistant bacteria is quite close in that biofilms, because of the presence of persister cells and being able to trade genes between each other, can often have an increased level of antibiotic resistance. And so I think moving forward, the type of technologies we need to develop and the type of research that we need to do needs to really focus on both of these problems together. Biofilms as one distinct entity, but also as antibiotic resistance and how they interplay with each other to cause human disease.
Now what this says is that clinicians have to be much more careful and patients have to be much more vigilant about being in a hospital setting, about having surgery, because these are all times and areas where one can pick up bacteria that can lead to biofilms.
Hi, my name is Richard Longland and I'm the founder of the Arthroplasty Patient Foundation. I'd like to share a sneak preview of a film I've been working on since 2008. It's an issue involving tens of millions of Americans, but it's never mentioned in newspapers or on TV. This issue is the biggest elephant in the room, perhaps even a herd of elephants. I'll explain why I became interested in this complex world of bacterial biofilms, get into some incredible statistics, then show you an animation in the film scheduled for release in 2011. I was debilitated in 2003 with severe lumbar disc disease and did my research and received artificial disc replacement. In my recovery, I created the ADR support community to help spine patients. I soon realized that some patients had a long list of comorbidities or health problems, along with their spinal pains. And many were desperate for relief and suffer with the "just fix me" condition that afflicts so many people in severe pain. I continue to see hundreds of spine patients experiencing severe complications ranging from MRSA infections to more complicated subclinical problems. These patients suffer in ways that are beyond description. So now, my role is multifaceted as a researcher, nonprofit founder, filmmaker, and especially a change agent for healthcare. Do you ever wonder why more than half of America is medicated? Or stated differently, why chronic disease is so prevalent in America? That's always bothered me, and it's just not right. In the meantime, let's focus on part of this problem, the facts relating to chronic bacterial diseases. There's no question that there are many different kinds of microbes that contribute to diseases. Viruses, bacteria, yeast, and fungi, to name a few. But as it turns out, many of these different microbes cooperate to live in communities within the human host. They're just doing what they've evolved to do. Unfortunately, they are creating a hellish suffering for us human beings. There are literally tens of thousands of research articles devoted to bacterial infections, with some of them published a century ago. However, most articles relate to Koch's postulates developed in the 19th century. As it turns out, these postulates don't necessarily explain the behaviors of bacteria in a bacterial biofilm community. Could our incomplete understanding of bacterial communities explain why so many patients are undiagnosed and untreated? Why hasn't modern medicine modernized? Actually, the notion of bacterial biofilms has been around since the 70s. Much of this was started by Dr. Bill Costerton at Montana State. These pioneers identified how bacterial biofilms enter and propagate within the human host, grow, avoid detection and eradication. In the 1980s, their research continued with meager support while millions of people continued suffering with chronic bacterial diseases. Thankfully, biofilm research and development continues throughout the world today. Currently, bacterial biofilms are better understood and accepted by academia, government, and the medical establishment. But the pace of change is slow, as evident by this strange fact. Dental students know more about bacterial biofilms than most physicians. But again, there's hope as incredible progress has been made in biofilm research. Case in point, let's take a closer look at how pervasive biofilms can be in the human body. As researchers now know, every part of the human body can host a bacterial biofilm community causing infection, as depicted by this graphic. One 2000 paper using data from 2005 on medical biofilms tabulates the following incidence of biofilm-related diseases: 550,000 deaths, $94 billion in direct costs, 17 million confirmed cases. These statistics were compiled across a number of different specialties, for example, neurological, gastrointestinal, urinary, dermal, etc. It does not include all specialties and unreported cases. Again, 17 million cases. That's 17 million human beings alone in the United States. Let's take a closer look at the differences between two different bacterial disease models. The first one is taught and practiced by physicians today. It's the classic acute infection model that uses culturing diagnostics to confirm a bacterial infection. This method is able to identify far less than 10% of all bacteria that could adversely affect humans. The second model is a problem and an opportunity, as it represents a new way of explaining how bacteria live subclinically in the human host. It requires that clinicians use new molecular diagnostics technology to identify chronic infections and new ways of treating and healing patients once the infections are found. This new approach is being practiced today in a small number of clinics and has been very successful in treating chronic wounds. I'll conclude with a preview of an animation that illustrates the bacterial biofilm growth process. It's simplified, showing the stages of a monomicrobial biofilm, and may give you a new appreciation for the complexities of bacterial disease. Thanks for taking the time to watch this important video. Pass it on to people who you know might be interested, and help America get back on the path to wellness. Thanks for your time. The life cycle begins when bacteria enter the bloodstream from various infection routes and evade the immune system or antibiotics. Formation begins with attachment of planktonic bacteria to the surface, preconditioned with proteins from the host immune system. Irreversible attachment to host proteins signals the bacteria to create biofilm and is the first committed step of biofilm growth. The bacteria anchor themselves permanently using cell adhesion structures such as pili and receptors called epitopes. This encourages new bacteria to attach to newly arriving cells, providing adhesion sites and building the protective matrix of the biofilm structure. Cells now communicate via quorum sensing and use small molecules to regulate gene expression within the bacterium. The community grows through both cell division and recruitment. The development of this complex biofilm makes the community resistant to the immune response and antibiotics. It may include diverse species of bacteria, yeasts, minerals, and host proteins. Recruited neutrophils result in a cascade of inflammatory cytokines and host proteases. These damage tissue and regulate fluid accumulation and exudate, providing nutrients. Bacteria now exist in multiple forms and metabolic states. Now a different phenotype, both physically and metabolically, it has the survival advantages of diversity and multiple defenses. The life cycle continues as fragments or planktonic bacteria are released to reinfect the human host.